From the campus of Stanford University.
People are worried about data. They're worried about their privacy and their security. They should be. We need secure systems.
This is the future of everything.
But we can't have a system that closes that data off. It is too rich of a source of inspiration, innovation, and discovery for new things in medicine.
With your host, Russ Altman.
Today on The Future of Everything, the future of genomics. Now, every living organism on Earth has a genome. This is the DNA molecule that encodes for how we develop from a fertilized egg, or a seed if we're a plant, to a full organism. And the physical features that we have, what we look like, how big we are, what our organs look like, as well as our risk for disease, and how do we respond to things that we ingest, both foods and drugs. Now, I think everybody understands that we inherit our genomic DNA from our parents, and so we share lots of our DNA with our close relatives and decreasing amounts of our DNA as our relatives become more distant, second, third, fourth cousins. I mean distant in our relationships, not in space. There has been an explosion since the year 2000 in our ability to measure DNA. It's a long molecule made up of 4 building blocks. The famous DNA bases, A, T, C, and G, are their nicknames. In humans, we can divide our DNA into chromosomes, long strings of these DNA, and 3 billion of them is what you get from mom, and you get 3 billion from dad. And you can get your genome sequenced. You can get the series of those 4 bases really for less than $1,000. In fact, I have a genome, my genome, in a hard drive in my office, sitting there. It has a password on it, and I taped the password onto the hard drive, so it's not the most secure genome in the world. Now, since 2000, when we sequenced some of the first genomes, there's been a blossoming in the use of genomes, both for humans, plants, and animals. For humans, we can understand and begin to try to predict disease risks. We can try to understand the basis for physical and biological traits. We can understand our ancestry, both out of interest for what our roots are, but also for forensic purposes. And there's been in the news recent cases where the relatedness of individuals has been used to solve long unsolved crimes. We can also study the movement of human populations based on who is similar to who. That gives you information about where somebody may have came from and where they went afterwards. It also has been used in animals for husbandry, improving the health of farm animals, improving the quality of meat products, and for pets, for dogs, for cats, in order to understand their heritage and also their risk for diseases. Professor Carlos Bustamante is a professor of biomedical data science, genetics, and biology at Stanford University. He is an expert on the use of DNA to understand both human biology as well as animal biology. Carlos, one area that you have highlighted in your work is that we have mostly studied the DNA of people of European descent, and not people whose ancestors might have come from Africa, from Latin America, from Asia. Why did that happen, and is it really a problem?
Hi, Ross. Thank you. It's great to be on the show, and it's exciting to talk to you about this topic, which you know is sort of my one of the most passionate things that I spend my time on. So I would say it's a little bit of an accident of history, and at the same time, I think it's a concern, but also an opportunity. So when DNA sequencing technology began to plummet,
The cost,
right? Yeah, sorry.
Capabilities went up,
right?
Cost
exactly,
plummeted.
and the cost of being able to create maps of genetic variation became accessible, right?
Yep.
The first places to invest were the European countries that saw the opportunity for this to impact healthcare. And in particular, one of the most successful places was Iceland.
Iceland.
Yeah, Decode Genetics got spun up. The Brits started a project out of the Wellcome Trust. And so they just invested quickly, and by the year, say, 2009, They'd done about 1,000 studies, and 95% of the participants in those studies were of European descent, be they from the countries in Europe or in Iceland.
And these
or
are
the
not
US.
bad guys. They're just Icelanders, and they go outside and
this is the opportunity, right?
Right.
So we called attention to this and said, guys, if data is the new oil, we've only been looking for oil in the North Sea. Right, there's tons of opportunities in other populations. We got involved with the 1,000 Genomes Project and called attention to this as part of that effort. They then expanded the 1,000 genomes to be 2,500 genomes, including 500 from the Americas that we helped collect.
Now, we hear that many humans are very similar, and obviously that we all see each other, and our hair is a little different color or different heights. But why is it that an Icelandic genome couldn't be or isn't as useful for an Asian descent person or a Latin American person? What are we missing by not looking at those populations?
Great question. So first thing to point out is, as you say, we're 99.9% identical at the DNA level, but it's the differences that may be important.
Because when you have 3 billion bases, even a 0.1% difference is
3
a
million.
lot of DNA that's different.
3 million differences between what you got from your mom and what you got from your dad. So let's just take a simple example. Let's say blonde hair, right? And you look at populations that have blonde hair. It turns out there are two main populations that have blonde hair: Europeans and Melanesians. And so Melanesians, people in the Solomon Islands, actually, they're these beautiful kids with blonde hair. And so when we started a project to try to get at the genetic basis of blonde hair, I was pretty sure that this was going to be a Captain Cook allele, right? You had
some European went to Melanesia.
Some European and left a lot of kids.
In particular, some blonde European.
Presumably.
Okay.
Right? And so when we did the genetics of this, it turns out, and it didn't need to turn out this way, but it just happened to turn out this way, that the genetics of blonde hair in Europe are different than the genetics of blonde hair in Melanesia.
Different causes.
Different genetic causes, right? They look the same, but it turns out that the genes underlying the difference are different.
Huh.
And why is that interesting? Well, I think first, from a biological point of view, it's kind of cool that blonde hair has evolved twice in human populations. From the point of view of medical genetics, if this is blonde hair, which is about as simple a trait, I mean, you don't need to be a Stanford-trained dermatologist.
You can make that call.
Right. You need to be about 5 years old, right, and look around and say, geez, some people have blonde hair, some people have brown hair, some people have red hair. Why? Right? One of the most simple questions you could ask. What about diabetes? Why would we presuppose that the genetic base of diabetes is the same in every population?
That was the jump.
Right?
So a Melanesian with diabetes might have an entirely different set of problems than a European with diabetes.
Absolutely. Well, from your training as a physician that diabetes actually presents differently in different populations, right? Lower body mass index in Asian populations and European populations, for example. Another neat example is the ability to digest milk. So it turns out humans are the only mammalian species that drinks milk as adult, right? You never saw an adult horse drink milk, right?
Yes, and in fact, that's a very discomforting image you just created.
Sorry about that in your mind's eye. All right. So why on earth do some people drink milk, right? It's kind of an interesting question. Turns out that, again, there are two main populations that drink milk: Northern Europeans, and there's some African pastoralist populations like the Maasai that also drink milk. And you can begin to think about it. Well, what's the
And when you say drink milk, you're talking about everybody can try milk, but there's a large percent of the population that when they try milk get incredibly bad gastrointestinal symptoms.
After you're so usually as a child, almost all humans can drink milk, and this is true of all mammals, right? The genes to digest milk get turned off in all mammals after weaning, right? Why on earth would you produce a protein to digest milk if you're not drinking milk? It's a waste of, metabolism. It turns out that there's some humans that carry a mutation that constitutively express this gene. That means it's always turned on, okay? And in Europeans, we've mapped that genetic basis, and it turns out to be the gene with the strongest signature of natural selection. And we can tell a story that during the last ice age, if you were living in Northern Europe and you could drink milk, then you have a higher probability of surviving than if you didn't drink milk.
Right.
Okay? Well, what about Africa? Those people that drink milk in Africa. Turns out they drink milk. It's the same gene, but the mutations are different.
And there was no ice age there.
And there was no ice age there. But they started to have a pastoral lifestyle where they also, have cattle and so on. So it's kind of cool, right? That as we've broaden representation in these kinds of studies, we've found interesting biology. And that's really in my mind, that's the reason to do it. I'd also argue that when we think about the opportunities for new drug discovery, okay, one of the biggest challenges we face is that bringing new drugs to market is extraordinarily expensive.
$1 billion.
More than $1 billion.
Right.
And part of the reason is that most drugs fail, right? You got a 1 in 20 chance of it making it all the way through the pipeline. It turns out that drugs that are based on genetic leads, about 30 to 40% of those seem to make it through the pipeline. And probably one of the best examples is a cholesterol-lowering drug called PCSK9 inhibitors. Those were found by studying families with naturally high levels of cholesterol, so-called hypercholesterolemia, familial hypercholesterolemia. And what's cool about that story is that there are two extremes of the distribution, those with naturally high levels of cholesterol and then those with naturally low levels of cholesterol. And these were people born with a Superman mutation. They could eat as many hamburgers and hot dogs as they want.
It's not a problem.
It's not a problem. And we can now mimic that.
What a gift.
What a gift. Imagine that. Eat as much pizza as you want. You're good. Right? And in fact, there was a woman born with 2 copies of the mutation. She was an aerobics instructor in Dallas, Texas. She had a cholesterol level of 15.
Where
right?
it's usually up above 100.
Yeah. Your doc wants you to keep it under 100. Imagine naturally 15. Gift from God. So, if we're going out and doing these studies, the point is if we spread our bets across different human populations, we're much more likely to find interesting biology that then benefits everybody.
Right.
Because these cholesterol-lowering drugs aren't just good for those people with high cholesterol for genetic reasons.
That's the key.
That's the key. You can mimic it in others, and it benefits everybody.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Carlos Bustamante about genomics and diversity. So, great. So we've established 2 things: that most of our current knowledge, for a bunch of historical reasons, as you pointed out, are from Northern Europeans. And then there should be a sense of urgency in the scientific community to sample the other populations. Yeah. Now, this is not a trivial thing. The one that comes to mind right away are Native Americans who have very firmly held beliefs about ancestry and the appropriateness of querying. So it becomes not just a scientific problem. So what is the approaches to sensitively approach these populations and make the case that you just made extremely compelling? Is it compelling to all populations?
Well, so here's the argument. I was born in Venezuela, as you know. A lot of our academic work has been in Latin America, and we've really been led by the mantra. The mantra in the lab has always been do the right thing. Right. So how do we go into these populations and try to engage them?
Exactly.
And one of the best examples, we have the work we did in Mexico, for example. So in Mexico, we were able to work very closely with anthropologists that had worked with Mexican Native American groups. We engaged those groups. They had a 10-year relationship with those groups that allowed us to come in and do probably the deepest study that had been done of the genetics of Mexican Native populations. It was fascinating.
When it goes wrong, I've heard it called helicopter genetics, where you take your helicopter, you come in, you get samples, you make all kinds of promises, and they never hear from you again. It sounds like this is not
this
what
is
you
the
did.
opposite of helicopter genetics. Right. I had a pair of postdocs in my lab who were from Mexico. This was what they were passionate about. They've actually gone back to Mexico. They've got the leading lab, in my opinion, for Mexican medical genetics. Yep. And it's been sort of
and then you could
virtuous cycle.
Win
absolutely
the engagement.
You win hearts and minds,
right?
And one of the arguments I make when I go, especially talk in Latin America, talked in Colombia, talked in Brazil, talked in Peru, I said, look, if you don't do it, no one's going to do it for you, right? This shouldn't be the job of the NIH to go out and study every population on the planet. Right? They've got a mandate to work in the United States, right? So U.S. minority populations obviously need to be represented, right? And then the rest of the world can pick up the ball in terms of their own responsibility to their populations. One of the things NIH has done is invest, for example, in Africa in a project called H3 Africa, where they're doing capacity building in Africa so that African scientists can lead this work. It's the right thing to do. Okay. So, and part of the argument I use is, look, if you don't participate, you won't benefit. Right? There's a ton of this information that relies on being able to really study these populations where these mutations have only arisen in the last 500 or 1,000 years. Colombia is a great example. There are valleys in Colombia where every valley is a little bit different than every valley next to it. And so, you're beginning to see lots of advances. Alzheimer's, for example.
And they might have, I think what you're saying is they might have variations that are particularly important for certain diseases, and we'll never know about them unless we get to see their DNA.
There you go. Exactly right.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Carlos Bustamante. You mentioned the importance of populations in the United States. And one of the things I know you've written about is some of the genomic, I don't know how I would put this, the genomic fingerprints of the history of slavery in the United States. And you've looked at this a lot, and of course, a very sensitive issue.
Yeah.
What are the things we can learn by looking at African Americans in particular? We know that they are a mixture typically of European ancestry and African ancestry. It's relatively recent, over the last few hundred years. What do we see when we do those kinds of studies?
Great question. I mean, the transatlantic slave trade is probably one of the greatest crimes in human history. 20 million people forcibly kidnapped from Africa over hundreds of years, brought to the Americas. A lot of that history has been lost, and African Americans want to reclaim parts of that history using DNA. Because it was an industrial enterprise, there were records kept. So we've got slave ship records that give us part of that history. What's interesting, at least in the United States, is that most of the slave ships went first to the Caribbean and to Brazil. Only a couple 100,000 people came in straight to the port of Charleston. So the history of the slave trade is actually written in the DNA of the Caribbean populations, the Brazilian populations, the U.S. African descent populations. So we've done a mixture of studying modern individuals as well as individuals from slave cemeteries. So we did a project where we looked at skulls from St. Martin's that seemed, from a bioanthropological point of view, seemed to be individuals of African descent. They had filed teeth, and the isotopes seemed to suggest as a person born in Africa, but they were in the Caribbean. They died in the Caribbean. And we were able to use DNA from those samples to trace the history back to particular tribal groups in Africa as part of a collaboration where we had gotten.
And I can imagine that would be incredibly inspiring to an African descent person in the United States who all they know is my ancestors were taken over on a ship and I don't know what. And now you can say, you know what? Here's an area of Ghana or whatever where your ancestors came from. And it can be in the same way that many of us who are European descent, I go to Ireland and I found the town where my grandmother was born, and that's a big deal.
That's a big deal.
And if you take that away from somebody, it's a big deal to take away.
Absolutely. And I'm very optimistic about this program for the reason that companies like 23andMe and Ancestry have now built extraordinarily powerful databases that are just compounding in value. And when you link those to historical records, in the next decade, we will have a pretty complete pedigree of everybody alive today and almost everybody who's lived and left a historical record, which is pretty wild.
And it really begins to really make real the human family.
Absolutely.
It's not an analogy.
No.
It's what is the truth.
It's what it is. It's a giant, it's the list of the begats from the Bible that we're now building from today all the way back. And I think that's extraordinarily cool because you'll be able to tie all those pieces together.
This is The Future of Everything. I'm Russ Altman. More with Carlos Bustamante about genomics and its multiple uses next on SiriusXM Insight 121.
From the campus of Stanford University.
People are worried about data. They're worried about their privacy and their security. They should be. We need secure systems.
This is the future of everything.
But we can't have a system that closes that data off. It is too rich of a source of inspiration, innovation, and discovery for new things in medicine.
With your host, Russ Altman.
Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Carlos Bustamante about genomes and their wide uses. Carlos, when I look at what you've written about, I see papers about lions, birds, horses, gorillas, and dogs. So let's go to dogs. You've written a
Man's
lot
best friend.
about dog genomics. Yeah. What do we do with dog genomes?
So dogs are incredible, right? We've relied on them since the dawn of time. And one of the big questions.
In many ways, we've
co-evolved. We've co-evolved with dogs, no doubt. And one of the coolest questions that we tried to understand is how do we use genetic tools to understand the vast variation that you see in dog breeds, right? Orders of magnitude in size.
Unbelievable.
First paper we wrote on this was this is a long collaboration with Elaine Ostranders and others, was the gene for dog body size. And we normally in biology try to say, there are no genes for, right? That's a no-no.
You should always have a red flag when the New York Times says they found the gene for X.
Well, I'll tell you, there is definitely a gene for small dogs. There's the yippy gene, okay? Every single small dog that we sequenced and looked at, any dog below 10 pounds, I will bet you $100 has a mutation in the insulin growth factor 1 gene. Okay?
IGF-1.
IGF-1, one of the most, the canonical genes in biology. And in fact, that gene goes back 10,000 years. That mutation goes back 10,000
Which is probably roughly when humans and dogs started doing their thing.
Well, that's the thing, right? So you can imagine, what are the models of dog domestication? You want to go out and try to wrestle a wolf pup?
Hey, wolf.
No, I'm good, right? But if you got some little wolfy dog that's hanging out on the outskirts of a human settlement, I'll let that cute guy stick around, and then he'll follow me.
As long as he doesn't bite me.
So it's the sort of self-domestication theory, right? And the dogs start following humans around, help us with
hunting. Because then they do get fed, and all of a sudden life can be pretty good.
Dogs are wonderful human commensals, right? They eat the same things we eat, right? So we began there and then layered on genes for coat color and coat texture. And in fact, the coolest applications, I think, are in the direct-to-consumer space. So about a decade ago, I helped the Mars folks spin up a dog DNA test called the Wisdom Panel. That was used to tell people what kind of mutt they had, right? So you take the dog, you take a swab of the dog's DNA, and then they can tell you it's a mixture of, Great Dane with Chihuahua and Border Collie. And
you get like percentages.
You get percentages. You get a whole family tree of your dog.
So there could be surprises.
There could be surprises.
We thought Fido was a bulldog, but he's not.
No. It turns out, no. Fido lied to you. I don't know you, Fido. And then what's cool is that the technology's gotten better. A couple of my folks that have been in my lab started another company called Embark that's now
Let's just pause for a moment. Okay,
it's doing beautifully well. I'm so proud of them. And so I'm really excited. There's sort of like this whole explosion of what we're able to learn that has a virtuous cycle, right? They've actually now found the gene, the Embark folks have found the gene for blue eyes in Huskies by looking at
bewitching. That's the only word I can.
I like it. I like it. I don't know if it moves its snout, right? So the long-term opportunities here, I think, are massive, right? You can imagine being able to use the dog's DNA and that database of dog DNAs to inform dog breeding and to inform how we keep dogs. Matt, you could get an extra 25%, 30% out of your dog's life.
Yeah, I wanted to ask, so you had initially kind of pitched it, so to speak, because of ancestry and what kind of mix, but I would imagine that there are dog health health issues.
Huge.
There are certain cancers. There are certain diseases. And I wanted to ask you, is this going to be something that pet owners who love their dogs might want to or need to do just to help the dog?
Well, today, right, we microchip almost every dog. I think in the next decade, we'll clearly DNA sequence every dog. It's, it'll be
unbelievable.
Cheap. It's just,
and what are the kinds of interventions that you can see on the horizon that a dog owner might take because of this genomic data?
Well, you can imagine perhaps you need to have more common, more screening for hip dysplasia or other disorders. You might be able to eliminate certain diseases from dog populations.
By selective breeding. Because that's the other thing is we worry that certain breeds have become so inbred that they start to have coexistent disease risks.
Absolutely.
But now there's an opportunity, I would guess, to say we can perpetuate this breed, but we can take away that terrible scourge.
I'll be honest with you. This is the future of everything. I also want designer dogs. Imagine being able to, order up the dog, and then we'll CRISPR them for you, and that's already happening.
Hold that thought. This is the future of everything. I'm Russ Altman. I'm speaking with Carlos Bustamante, and we're going there. We are going to clone your dog. So
no, that dog cloning is already a thing.
So
that's been around for a decade.
Tell me about that.
Right? The biology has had that biology has been worked out. One of the toughest problems in the dog engineering side is that
dog engineering.
That in vitro fertilization in dogs turns out to be really hard. The egg of the dog, you just got to penetrate it with like a steel syringe.
So we can do it for humans, but it turns out that it's tougher for dogs.
It's tougher for dogs, but it's working. I mean, people are getting it to work. I used to be at Cornell. Cornell vet's very interested in this problem. And I think, again, there's a massive interest in this space. And if you bring enlightened capital to bear.
Because you like Fido's personality, but Fido's getting old. Let's get a new Fido.
Well, plus, or maybe you live in New York and you really like a Great Dane, but you want like a 30-pound Great Dane.
So we do the IGF-1
We do
trick.
the IGF-1. You make them small. Well, in fact, there
So
are
it's not
about
such a Great
10,
Dane.
There's about 10 genes that in total explain about 80 to 90% of dog body size variation. So I can get you. You want a giant Chihuahua? I'll get you a giant Chihuahua. That'd be crazy, though. Who'd want a giant Chihuahua?
So what you're referring to is there's now been this revolution. It's CRISPR. Everybody's read about CRISPR. It's a way to edit the DNA.
Yep.
Because of the studies of you and others, we know which genes matter for which traits. So it really becomes possible to create an engineering discipline.
Absolutely.
Where you say, how would you like your dog? So we can start with a substrate of a dog that you like, but there's a few things that you don't like. And is this going to happen, or am I just making up fiction?
No, this is already happening. And I would argue that some of the first things that we saw coming out of China in terms of CRISPR were actually in dogs. We had mapped years ago a gene that was involved in musculature in dogs. So we had looked at whippets. And whippets are like a Great Dane, but smaller.
Yes.
They also they run the races.
Yes.
And there was a bully whippet phenotype, which was these
I've seen pictures
double
of this dog.
muscled
Yes.
dog that looked like Belgian cattle. Turns out they had a mutation in the myostatin gene. And the reason that mutation stuck around is because of the heterozygote dogs, the ones that had one copy, looked like a super chiseled, and they ran faster. Right. So those elite dogs.
And that was the selective pressure.
That was the selective pressure. They've now cloned that into other dogs supposedly.
So you can get
I don't know if it's fake
a
news or
super
not.
muscular Chihuahua, maybe.
If you wanted one. So, and I would say the other cool application that we're seeing, aside from dogs, is in horses. So there's a horse genetics company that we've helped get off the ground. They're trying to do something very similar, understand the genetics of the horse, largely to, again, improve the quality and the lives of our companion animals.
That's quite important because I don't want to be glib about, you don't want to cause suffering in these animals.
No.
And so I'm sure that people, or I would certainly hope that the people who are engineering these animals are thinking about, we can't have too much suffering. We can't give them traits that cause them to have a life.
Absolutely. Well, I mean, look, we've been doing selective breeding in plants and animals since the dawn of agriculture, right? Now this is a separate an additional toolbox that we can bring to bear. And marker-assisted selection has proven to be extraordinarily useful in improving yield and improving many of the qualities we want in plants. That's now being brought into the animal realm. There are, of course, ethical issues that need to get
you
brought
could
to bear
imagine
on here.
that the experience we get with dogs and horses, you can't help but imagine will inform potential future human applications. And of course, because of some activities recently in China, this is coming a lot sooner than anybody really thought it should. But there is a certain logic to saying, let's make sure we can do it well in our fellow animals before we start messing with humans.
That's always been the way science proceeds, right? You often need to test things in animals before you do it in humans. And again, I'm optimistic that we'll be able to settle on a set of technologies and that there'll be a marketplace that supports this with proper regulation and sort of guidelines and guardrails. But it's extraordinarily exciting because when I went to graduate school, there was no human genome, right? The idea that you'd go study the statistics of DNA was a terrible idea. People told me, don't do that. There'll be no jobs. And then now we've got millions of sequenced human genomes, tens of millions in the databases from the online companies, and in a decade, it'll be hundreds of millions. So it's an extraordinarily exciting time. Encourage the young people out there to consider a career in genetics and data science. It's been extraordinarily exciting for me and for you, and I think the future looks very bright in that regard.
Thank you for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app.